Hi, this is Bill Fitzgerald, Vice President of Growth and Ecosystem for SandboxAQ, coming to you from the Voices of Davos. Very excited to speak to Franck Zal from Hemarina, an incredible organization that is driving forward novel therapies. And I'd love to hear, Franck, a little bit about the business and what you're here for.
Thank you so much first for your invitation, Bill, and thank you to Sandbox to give me the opportunity to present Hemarina. So I am the CEO of a company called Hemarina. It's a biotech company. I have created this company 20 years ago. My background, I am a marine biologist. I did during 15 years research in academic position, and I discovered a molecule which is a big molecule, which is a polymer coming from marine worm, the worm called Arenicola marina, or in English, lugworm. And we extract this molecule, and what we have discovered in this molecule, we have discovered the ancestor of all red blood cells. This molecule is able to deliver oxygen to the level of cell, tissue, organ, or full organism. So you can use it for organ preservation. We have a product called Hemo2Life in order to keep longer time the organ. For example, to pass some hour to some days. So it changed everything. It's not an emergency anymore, and you deliver organ in very good condition to the physician. We have a product for dental care, for periodontal disease or peri-implant disease, to kill the anaerobic bacteria that you can have between the gum and the teeth. And so this one is in dental care, but we have a product in transfusion called emoxy carrier. Imagine tomorrow blood in powder. So we are able to make blood in powder. When you provide blood, when you give blood, the blood donation, you can keep it during 42 days at 4 degrees Celsius with blood typing. And after 42 days, you need to remove this one in the circulation because there is some hemolysis and so on. We are able to have 5 years at room temperature of blood in powder without blood typing. So it's a revolution. And we are developing other products in oncology for glioblastoma, in ophthalmology in order to Fuchs disease. And it's not there is not an end just because we know how to manage the delivering of oxygen at all the level of life. So it's like a breakthrough innovation. We have a bench of collaboration worldwide, and we're just starting one, with U.S. Navy, with the blood in powder and the bacteria that we can find in battlefield. And you heard about the gases gangrenes that's coming from people suffering of injury in battlefield in Ukraine. So we start this collaboration with the people in Maryland, with the Navy, the research center of U.S. Navy.
Excellent. Franck, what you just described is an amazing application of the molecule across a variety of different simulations, a variety of different deployments across the body. Tell me about how you think about AI and simulation in that field.
In my point of view, AI will change everything. Today, when you develop something from scratch, you have over 90% of failure, just because you need to pass preclinical phase 1, phase 2, phase 3, and 90% fail. You can spend $1 billion and to fail at the end. So if you can simulate it, the preclinical package with all these kinds of the same stuff, again, cytotoxicity, genotoxicity, biocompatibility by AI, it would be very wonderful. The only request that I have for you, Bill, it's how it will be regulated. If I come with this data to the regulatory people, they told me, it's fine, it's work. Yeah. Go to phase 1. You don't need to do a preclinical test. Yes. If it's okay, all people going to use AI to make this kind of package.
Yes. Yeah, and I think inherently in organizations like Sandbox, we have an obligation to practitioners like yourself to increase the amount of explainability in the models that we create together. So you have the burden of having to prove the science and the compilation of steps that got you to the output of that science. We should have that same obligation. And at Sandbox, given that our LQMs, our large quantitative models, are grounded in physics and chemistry and mathematics, as well as the capability to deliver a knowledge graph of where contextually all of these components fit together, that we think we can be a great partner in helping to achieve the development of regulation as we go through this and help you, bring that story that you so desire.
Yeah, sure. And Bill, you know what we can do? We can make partnership. I give you the data, and we make reverse. So I can provide you all the data, cDNA, quaternary structure, and all the data on toxicity. And you will manage all this data, and you told me, okay, I can predict it will work just because we have this point of connection.
Yes.
And it will be a wonderful tool. And believe me, most of the people who are developing drugs, they hate, believe me, they hate to kill animal.
Yes, yes. And I think, what you described earlier was a translational science that doesn't always, translate from the animal testing into the human as is. That's a current problem in drug development.
Exactly.
So if we could successfully simulate at the molecular level that the way the body is going to react across a variety of different therapeutic possibilities, we could actually get more accurate. And the best way that I know that is to do work with great partners like yourself. So thank you, Franck.
Thank you so much
for everything you're doing. This field is super exciting. Certainly the stories of the potential for blood to be delivered on the battlefield in a more sustainable way, in a more impactful way. We wish you the best with the work you do in the company, and thank you very much.
And thank you again, and thank you for your invitation.
Absolutely.